I'm pleased to welcome Dr. David Fike as our Okubo Scholar, and Jack will be providing more information about him for his introduction shortly after I'm done. But I just wanted to provide a little bit of background for those of you who might not know about what our Okubo Scholar Symposium is. So our Okubo Scholar Symposium happens every year, and it's a tribute to a professor of mathematical ecology that was here at SoMAS named Akira Okubo. He was a distinguished faculty member. He was very beloved here. He died of cancer in 1996. So after he passed away, we've had this symposium to honor him by bringing in a distinguished faculty member from another university to speak about their research and their career journey, which we'll be hearing about here today. So I'll turn this over to Jack for David's intro.
Sure. So as Madison said, this year's esteemed Okubo Scholar, Dr. David Fike, is a distinguished professor and chair of Earth, Environmental, and Planetary Science at Washington University at St. Louis. With a doctorate from MIT, Dr. Fike's research has spanned a wide variety of topics pertaining to sedimentary isotope biogeochemistry, with the aim to understand past and present biogeochemical cycles, more specifically including sulfur, iron, and carbon cycles. You should see the list of papers and from him and his students. It's kind of overwhelming scrolling on his website. So without further ado, on behalf of the faculty and students here at SoMAS, I welcome Dr. David Fike.
Hi, everyone. Thanks so much for the invitation here as the Okubo visiting scholar. It's a pleasure to be able to interact with the students and faculty. I think I was here last many years ago, and so it's really great to be able to see everything that's going on right now in the school. This talk is kind of interesting. So I was asked to do sort of a career trajectory talk, and that's very different from a typical science talk. And so it was a bit of a puzzle and also an enjoyable experience to try to pull this together. So I hope you'll bear with me. This is supposed to be pretty informal, so please, if you have questions or comments throughout, I would much rather this be sort of a dialogue rather than me just sort of one way talking through things with you. So anyway, here we go. I am an isotope biogeochemist. I'm interested in using isotopes to understand the Earth, how it works today, and how it has changed over Earth history. I'm going to try to walk us through how I got from how I got here. All right. So I like maps. I've always liked maps. So this is a good map. I grew up in sort of northwest New Jersey, out in the boonies, and then moved to the Chicago suburbs through high school. I did my undergrad at the University of Illinois, and I started out in engineering and physics. And so I was working on problems like fluid dynamics. So this is a much younger picture of me. We were looking at sort of rotational fluid dynamics, and we got to fly in NASA's KC-135, the little vomit comet that goes up and down, looking at how gravitational forces influence different things. And then I worked on things like particle physics, collisions, and chemistry of globular clusters using some of the Hubble data. And I was very excited about sort of astronomy and physics. And then I saw lots of my colleagues sort of go on into graduate school in this area where they were mostly sitting in front of a computer all day modeling. And that wasn't really what I was looking for. So I also did a project sort of on solar shields for NASA. There was a talk by a geologist in the astronomy department that talked about the KT impact and the extinction of the dinosaurs. And so the astronomers were interested in this because this was, thinking about comets and the intersection of comets and planets. But listening to this professor talk, it was like, oh, he basically gets paid to travel the world and go camping, and that was quite appealing to me. So I connected with him and did a senior thesis looking at hot springs in Yellowstone and trying to look at mineral microbe interactions and sort of how microbes influence the precipitation of minerals. And associated with that, this is black band disease in corals, trying to understand again the microbe mineral interactions and how microbes influence carbonate forming organisms. And then as a bit of an aside, to go back to the planetary side of things, I spent a summer at NASA working on a project trying to characterize Gusev crater on Mars. And that's where I got a little bit into the planetary side of things. And at that point, I sort of thought I wanted to do planetary science for my future. But before doing that, I sort of took a detour to Europe. I spent a summer at an archaeological excavation in Tuscany, which sort of started a lifelong love affair with the history and the culture of Italy. And now I do, coincidentally, a fair amount of my research in Italy. And I studied abroad in Darmstadt for a year, looking at some of the high energy physics side of things. I then went to Cambridge to get a master's, and I was still thinking on sort of a planetary focus. So I went there thinking that I was going to do science, but I also had a sort of Harry Potter fantasy, and so we got to dress up in robes and go to class like that. So that was pretty fun. I was at the Scott Polar Research Institute, and I was supposed to go and study in the British Antarctic station and think about sort of life in extreme environments and analog to maybe exploration of Mars. And then, unfortunately, right before we were supposed to go there, the British station had a severe fire and they had to cancel all the science plans. And so it's somewhat ironic, but Antarctic, there are a number of fires that do break out. And so it shut down the station, and I ended up switching research trajectories to work in the Canadian Arctic instead. This is Devon Island in northern Canada and the Houghton impact crater. And I sort of cobbled together a master's project that focused on microbial colonization of these sort of impact shock rocks. And it was really trying to understand how the volatilization associated with impact releases nutrients and creates space for microbes to then access those nutrients. So again, it was sort of with an eye to thinking about maybe astrobiology or life on Mars, early Mars kind of analogs. And so I was supposed to go then to the University of Arizona to do a PhD in planetary sciences. I had applied and I had been accepted, and then I put that on hold to go to England. And I was going to study sort of in situ resource utilization on Mars, like how you use the regolith on Mars and chemical reactions to try to extract hydrogen and other things. But that again didn't quite work out. And I think one of the threads that links together my sort of career path is serendipity or happenstance. And so while I was in England doing my master's, John Grotzinger, who was a professor at MIT, came through to give a talk, just a normal colloquium. And I met with him for coffee and we had a conversation and he said, hey, you seem interesting, you seem smart, why don't you come work for me? And I said, well, I'm supposed to go to Arizona to do this stuff. And he said, you can do that later. Why don't you come do your PhD with me? And so then I went to MIT. And one of the agreements with John, and John is a sort of a field geologist. He's a carbonate field geologist who made his name, looking at the Precambrian carbonates and stromatolites. And I said, okay, I'll come work with you, but I really do love planetary sciences, and I'd like to still get my foot in the door somehow. And so this was shortly before the Mars rovers Spirit and Opportunity landed, and John got involved with that, and then he sent me to work at JPL for that mission, and that's where I got to meet Scott McLennan, Joel Hurwitz, and so there's a lot of serendipity sort of associated with that. But the vast majority of my time at MIT was focused more on traditional geobiology. And based on my undergraduate work, I came in and I was convinced that I was going to do my PhD thesis looking at hot springs travogen. I had dabbled on that as an undergraduate at Illinois, and I said, hey, this is the thing that I get all fired up about. And John, to his credit, said, if that's what you're interested in, go for it. And we went out to Yellowstone, and I worked on it for about 6 months, and then I decided that's not really the thing that I was passionate about. There wasn't a question that I could really grapple with. And so I went into John's office and I said, I think I need to do something else. And he said, okay, what are you going to do? And I had spent a summer at NASA Ames working on a proposal to study high altitude lakes in the Andes. This is Lake Okubo in Chile. And that proposal got funded. And so I went with a team there and we spent about a month trying to characterize the geobiology of this high altitude lake, mostly looking at things like organic biomarkers, trying to look at the microbial community composition as an analog for early Earth, because we were up at really high elevation. So you had lower oxygen, you had high UV flux, trying to see, hey, are there differences in community composition associated with this lake versus what we see at lower elevations? And we went through a series of lakes at differing elevation as we sort of marched up the Andes. And that was really interesting. But after 6 months or so, I sort of couldn't really make a connection to the fundamental questions that I was interested in. And John said, okay, well, here's an idea. Why don't you work on a project that I'm already doing? And he had just started working in Oman, the Sultanate of Oman, on the end of the Arabian Peninsula, which has this beautiful package of Precambrian, Cambrian rocks. And again, he's a carbonate guy, so he was interested in mapping the sort of depositional facies of these carbonates, and that wasn't really my thing. I didn't really understand why that was interesting or important. Now I spend a lot of time, as Bob knows, thinking about these things, but at the time I wasn't into that. I was really interested more into the bigger picture environmental questions, and so we worked out an agreement where I would try to use geochemical approaches to figure out, this is the time period where we see animal fossils first appearing, and then the Cambrian explosion giving rise to sort of modern animals. And could we find any sort of geochemical evidence for a change in oxygen conditions during this time that we could plausibly link to the environment? And the approach that I decided I would use was to use stable isotopes. At this time, I had no real experience in stable isotopes. This is a classic figure from a Bob Burner paper who really sort of championed the idea of using carbon isotopes and sulfur isotopes. This is here for the Phanerozoic, looking at the record of carbon isotopes through time, sulfur isotopes through time. And Bob used this to sort of reconstruct a history of both CO2 and oxygen concentrations through the atmosphere. Noting that these tend to go in opposite directions, suggesting a sort of relationship there between carbon and sulfur regulating the oxygen in the atmosphere. And so I really like this idea. I liked, with my background sort of in physics, I liked the idea that chemistry could be able to tell us what was happening. That to me was more concrete than the kinds of things that John was doing, looking at depositional facies and trying to reconstruct microbialites and grainstones. This was a lot harder for me to really appreciate. So we got on board with this, and I went to Oman. This was a camel that basically followed me around for a couple of weeks while we were doing field work. And what I ended up studying for my PhD was this is the Ediacaran period from 635 to 542, which has this very pronounced carbon isotope excursion at this time that we argued is related to the final oxygenation of the ocean. And that seems to be linked to some profound changes in the development of bilaterian fossils. One challenge with that is I needed to learn isotopes to do this, and John didn't measure isotopes. But one of the nice things about John's approach to graduate school was he knew other people that measured isotopes. And so he sent me to Denmark to work in Don Canfield's lab, and then to Indiana to work in Lisa Pratt's lab. And I was able to learn how to make these isotope measurements that then became the sort of bread and butter of my own career. And so the thing that appealed to me about stable isotopes is that different kinds of biological processes, they discriminate between these different isotopes in a way that can be diagnostic for different metabolic pathways or environmental conditions. And then thinking back in terms of trying to reconstruct changes over the rock record, these signals can get preserved in organic matter that becomes part of the sedimentary record. They can get preserved in carbonate minerals and sulfate minerals and pyrite. And so we can use the isotopic compositions that get locked into the rock record as a way to tell these stories. And I got particularly excited about using sulfur isotopes. Sulfur has 4 stable isotopes. You can do really cool things with the minor isotopes, but the vast majority of work is done looking at 2 major isotopes using this sort of standard delta notation. And so that was how I really got traction into trying to interrogate the environment. When I was done at MIT, I ended up going to Caltech to do a postdoc with Victoria Orphan. And that was also this case of serendipity. When I was a first-year graduate student, I helped TA a geobiology course on Catalina Island that the University of Southern California was running. It's now being run out of Penn State. It's still an amazing course every summer for grad students. Victoria Orphan was one of the guest lecturers for a week. And we went kayaking together and we had a great time. And when I was graduating, she said, "Hey, I remembered you. We kayaked. Why don't you come do a postdoc with me?" And so that's how I ended up going to Caltech, and those kind of personal relationships you meet through conferences and visits to other schools really helped shape my my time. Now, Victoria is a microbial ecologist, so she and John were almost diametrically opposed ends of things. You know, he studied carbonate platforms that were, you know, thousands of kilometers long, and she studied, you know, these individual microbial cells. So that was a big culture change for me. I knew very little about the actual microbiology of how these cells functioned, and so I got to have a very steep learning curve. And I worked on these kinds of systems. These are microbial mats, in this case from Baja, California. This is kind of like the microbial version of lasagna. You know, it's this beautiful layered structure that is appealing in the sense that, because you can kind of imagine that it's really sort of you can do a one-dimensional profile, and you can see these different colors represent different groups of microbes, photosynthesizers, sulfate reducers, purple sulfur bacteria, green sulfur bacteria, methanotrophs, and how they're all metabolizing together. And so I was really excited to try to use isotopes to understand this kind of system. The challenge here, though, was, you know, when you're dealing with sort of big carbonate platforms, you can take a hand sample and maybe that's representative of the system that you're trying to figure out. We didn't really have a good way to make isotope measurements at the scale that was relevant for these kinds of microbial systems, right? This is two millimeters per scale. We really needed measurements that were, you know, down to hundreds of microns or even down to the micron scale. And the goal for me was, again, with an eye to the rock record, was understanding the sort of microbe environment interaction that was happening. That's what Victoria was interested in. But, you know, these microbial mats also get lithified. You can still see the sort of green pigments here and the purple sulfur bacteria here. And then this is one of the microbial mats that I actually looked at for my PhD. So this is a 540 million-year-old microbial mat that's preserved in anhydrite. And then, are there isotopic signatures that get preserved there that could tell us something about that environment? And so this is an image that we collected as part of this work. This is a fish image, so fluorescent in situ hybridization. The purple here is photosynthetic autofluorescence, but the green are sulfate-reducing bacteria. And I was particularly interested in those because they produce hydrogen sulfide, and there's a huge isotopic fractionation between the sulfate that they start with and then the hydrogen sulfide that they produce. And so I figured if we were going to use an isotope system to try to understand these settings, sulfur isotopes would be the best way to do that. These are just pictures of some of the different sulfate reducers that we see in these systems. And so we came up with an incredibly low-tech way to do this, which was we took one of these cores of this microbial mat. Again, you can see this nice laminated structure. And we just stuck a silver disc into it, like a silver coin. And silver really sucks up the sulfide to form a silver sulfide mineral, which is why silverware tarnishes. And so this is what that little disc would look like after we stuck it in. This would be the microbial mat surface, and then this is the zone of sulfide accumulation. You get sulfide accumulation with depth. We then took this coin and then stuck it in a SIMS instrument, a secondary ion mass spectrometer, and we were able to analyze the isotopic composition of the sulfide at those little points. And so this was probably the most expensive way we could think of to basically clean sulfur, but you can see that you ionize it and it comes off here. And what we could do is we could just go through and do a time series of sort of reconstructing the spatial variation here by just going up and down which just generates, a long series of individual measurements that we had to calibrate with some known standards. And then we could reconstruct that series of analyses into a spatial map of isotopic variability. So this is color-coded isotopes that if you were to rotate it this way would reflect this sort of grid here. So you can see that there are very clear stratigraphic patterns within these individual layers of the microbial map. So these are like, a couple of 100 microns in length scale here. And this was really exciting because we now had a way to make isotopic measurements that were relevant to the microbes. And in terms of a spatial scale that was diagnostic. And so these microbial mats go through day-night cycles of producing oxygen and consuming it. So the chemokline moves up and down every day. And we were able to generate isotopic profiles that match that. And you can see that they sort of move up and down each day. We get the same isotopic profile, right? And so we were able to try to constrain the processes that these microbes are creating that give rise to these patterns. And one of the things that we kept noticing is this persistent co-occurrence. These are large cyanobacterial filaments. You see sulfate reducers sort of coiled around them with the idea that perhaps those sulfate reducers are feeding off some of the organic matter that's produced by the cyanobacteria. And we see this isotopic enrichment for the chemokline, both in the day and the night. This is consistent with the idea that you get faster rates of sulfate reduction that happens in those environments, which causes smaller isotopic fractionation. So we could use this isotopic signature to fingerprint changes in the microbial population. And so after that time at Caltech, I started my faculty job at WashU, which was January of 2009. It's hard to believe. It's been about 15 years. And this is how I like to think of the key parameters here for geobiology. When I was doing my PhD, we were really trying to understand carbon and sulfur cycling on the sort of global, the planetary scale. So hundreds of millions of years over the whole planet. And then working with Victoria, we were really trying to understand, real-time isotope changes at the micron scale. And I've really devoted my time at WashU to try to link together these different parameters and really flesh out this sort of phase space of intermediate spatial and temporal scales. And focusing on that, my lab, we really have three different areas of interest. One is continuing to understand how environmental conditions, oxygen levels, pH, CO2 levels, sulfate abundance, how those regulate the types of microbial metabolism, the rates of microbial metabolism, and the geochemical signatures that metabolism creates, and how those in turn alter ambient chemistry. And a lot of that work is sort of built on the kinds of techniques that I learned in Victoria's lab. We've often gone to much lower tech approaches. This is Bill Gahuli, who's now faculty at Indiana University, Indianapolis, IUI. And this is Lake Mahoney in British Columbia. And instead of using the silver discs, what we did is we just took photographic film and we stapled it onto a 2x4. So this was a much lower tech version of the original thing. One of the cool things about Lake Mahoney is a stratified lake that has a sulfidic zone about 7 meters deep. And associated with that sulfidic zone, it's got this huge burst of purple sulfur bacteria. And they're so abundant that when you do a syringe pull You get this beautiful purple color, like it looks like Kool-Aid. And you could see these purple sulfur bacteria, their metabolic strategy is they take sulfide and they combine it with life and they oxidize it. And they were living just above the chemocline where we had sulfide accumulate, then you go into the oxid water above it. And so we were able to use that same kind of approach where we could then take the isotopic composition of the sulfide that we tracked and relate it to the microbial community that was present in the water column. And we've done this now in a variety of different environments. This is methane seeps off the coast of Costa Rica. These are microbial mats growing on the sea floor. You get this sort of circular concentric ring structure. And again, we would take cores, we put this photographic film in these cores, sulfide would template out onto the film, and then we could analyze that. Using the same kind of approaches, I don't know if any of you have been to Deciphering Salt Marsh. This is on Cape Cod. They have these amazing purple berries, but these are these consortia, again, of photosynthesizers and purple sulfur bacteria. And you could just pick up some of the mud and you see they've got these bright purple consortia on them. Sometimes they can get up to a centimeter in size. And so we were really interested in how do these consortia metabolize? What is their strategy? Why do they come together into these large consortia? And so we took a very similar approach. We got some silver wire and we just threaded these microbial balls onto this wire, templated out the sulfide, and then was able to analyze the abundance of the sulfur and the isotopic composition. You can see these sort of concentric patterns across the diameter of these berries. I should say it was very interesting to order all the silver for this because we had to go through like jewelry suppliers, and the university kept asking why we were buying, jewelry with our grant dollars. A little bit of an awkward conversation. But again, this idea that spatial isotopic values could provide insight into microbial ecology and microbial activity. And we see again this enrichment in isotopes toward the chemocline here. And this is exactly what you would expect if, say, disproportionation were happening, which is a sulfur splitting of intermediate valence sulfur compounds to reduced and oxidized components, which people have long thought is a key process in these kinds of settings. Another key aspect of our work is trying to understand how depositional conditions influence the geochemical signatures that life creates, how those depositional conditions sort of modify those or regulate which of those signatures get into the rock record. This is some Florida Keys, and I'm going to highlight, for this, some of the work that Bob has done. Looking here, this is off Papua New Guinea, looking at fairly shallow sediment cores, and we were interested in, okay, what is the pyrite isotopic composition that gets preserved in these? Is there any relationship between the water depth of these different cores? And this is just the depth. And one of the things that we really saw was in deep, we're going to use deep in a relative sense here. So 50 meters is going to be deep here. So like in terms of an ocean, that's incredibly shallow. In terms of this is deep. The pyrite isotopes were, for the most part, depleted around -30 or so, and not terribly variable. They were relatively uniform, and they looked like there was maybe just a continuous process, and then there was enrichment as you got down with depth due to distillation in the pore waters. And that was in marked contrast to what we saw with the shallower settings where the mean value was shifting. And there was also a lot more noise. And we could think about the mean value shifting as a result of change in sedimentation rate, regulating maybe the diffusive exchange between pore waters and the overlying water column. But to get to this kind of variability, you need to have other processes, probably like erosion and redeposition. So non-steady state processes that people don't often think about in deeper waters. But these are really important to consider generating these kinds of signatures in the shallow rock record. And again, we tend to have a preservation bias because of plate tectonics, so that if we're trying to reconstruct things in deep time, we usually only have the shallow records. And so the dependence of depositional conditions here really influences the signals that get preserved. Both of these settings have the same sulfate reduction happening, and they almost assuredly have the same isotopic fractionation during sulfate reduction. So there's probably no difference in the biology of the isotope discrimination. And this difference is due entirely, I think, to how depositional conditions regulate what gets preserved into that rock record. And the third major focus that we have is after deposition, how do signals get altered? How can we think about, if we have a rock from the Precambrian, how do we take the information and assign primary environmental values from it? How do we map out what is due to alteration and screen out for it? And this is a thin section through an Ordovician brainstone just above the base of the Hernanchen glaciation and mass extinction. You can see this sort of heart-shaped fossil would have been sort of spiraling in and out of the section. Right, and so here the idea is, this is a typical sort of Paleozoic carbonate. These things are quite complicated. They're made up of multiple different components, and those components all have their own chemistry and their own diagenetic history. This is just a example of some of the diversity we have for sort of thin sections of carbonates made up of multiple components. And I sort of tried to draw a cartoon here of, maybe you have something with an uid and some bioclasts in it. It gets deposited. Maybe you get some early marine cements that form. You get some maybe micronization, dissolution that happens. Later stage calcite forming. This is what happens to these rocks before we get them to analyze. And then what most geochemists tend to do is they just crush that up and they extract it, in a beaker. And all of the information that's present in all those different phases gets averaged out or integrated together. And depending on the questions you're asking for, that's probably not a good idea. So just as one example, we went in with this sample and we tried to map out, okay, thinking again on the sulfur side of things, how are sulfur phases in this carbonate varying across these different phases, early and diagenetic? And one of the approaches that we use in my lab is to take advantage of synchrotron-based X-ray approaches. This is Argonne National Lab. One of the key things about Argonne is it's a giant ring and you get to ride these amazing tricycles around, going from different sides of the facility. We're looking here at sulfur zanes, and the power of this technique is for different redox structures of the sulfur phases, you get a shift in the energy of this transition. So pyrite, for example, has a peak that's much lower down than the sulfate peak. So we could use the response of these different samples to constrain what type of sulfur moiety was present, whether we're dealing with an oxidized or a reduced phase or some intermediate component. Right, so here again, just is one example. Looking here, we have sulfide, we have sulfate, and some phases that have both the sulfide and the sulfate component. So we could use these approaches to map out not just where sulfur is, but where sulfate is, or where sulfide is. So again, going back to this kind of thin section, we can make a map of where the sulfides are. So distributed within this section, you have all of these sort of point sources. These are individual pyrites that have formed after that carbonate. And then this is looking at what the sulfate signal looks like. And you can see all of this late stage calcite has essentially no sulfate in it. So it's forming from pore waters that have been chemically evolved from seawater such that there's no more sulfate present. Whereas the grains themselves have varying amounts of sulfate in that those grains that have been microtized tend to have a lot more sulfate preserved as a result of maybe that microtization process. We can then link the changes in the sulfur to changes in other elemental abundances to try to reconstruct that sort of chemical evolution of these settings and understand not just where sulfur is or isn't, but maybe what are the processes that give rise to its change. And so now we're trying to sort of integrate these various approaches to think about biogeochemical cycling. And I want to sort of provide maybe two contrasting worldviews. This is what I would sort of call the inherited burner worldview of sulfate is in the ocean. Sulfate either leaves the ocean as sulfate minerals or after microbial sulfate reduction, forming hydrogen sulfide, reacts with iron to form pyrite. And then you use, very simple box modeling, taking advantage of this isotopic difference between these. And you can have a system where this is the fraction on the x-axis of what fraction of the sulfur leaving the system is that pyrite. This side, all sulfur will be leaving as sulfate. This side, all sulfur is leaving as pyrite. And then this is just a sort of statement of isotopic mass balance. So if you make a measurement of a sulfate or a sulfide, it can tell you something about the relative flux of oxidized versus reduced barium. And this kind of framework assumes implicitly that the values that one measures reflect the global ocean. And this goes into all kinds of compilations. This is one from Ben Gill looking at sulfur versus carbon and reconstructions of sort of global redox changes that happen. This is the basis for a whole suite of papers from Berner's classic papers to ones on the spice and the Cambrian, OAEs, and a couple of doozies looking at the Ediacaran Ocean. I'm going to argue that this approach is fundamentally flawed. And these interpretations are probably wrong because most of the sulfur phases that we see are forming in the sediments. And they're forming in the sediments where we have sulfate reduction that pulls down the sulfate profile and generally produces sulfide. Because of the isotopic fractionation during sulfate reduction, you get the evolution of pore water chemistry. And this is what gets preserved in the rock record. You're going to get values that are diagnostic for your particular environment, whether you're shallow or deep, rapid deposition or slow deposition, and not something that tells you about global ocean chemistry. And for me, this was a really important revelation. It's, I think, Bob, you would say, still not wildly popular with a lot of the isotope community out there. But I think it's right. And it goes against the things that I argued in my PhD. But as you get more data, you have to reevaluate, your worldview. And I think this fact that sediments are complex is really underappreciated because it has huge implications for how you think about the geochemical signatures that get preserved. And I want to just highlight one example that I will sort of beat to death tomorrow. But looking at this small scale isotopic composition or elemental composition is really critical to trying to reconstruct the bulk values. And the way we do this is with the secondary ion mass spectrometer approach, where you put your sample in, you bombard it with primary ions, either oxygen ions or cesium ions, depending on what elements you want to look at. These release secondary ions that you get. You can focus onto a series of collectors, and you could do anything from hydrogen up to uranium. Perhaps not surprisingly, mostly do sulfur, nitrogen, carbon, oxygen. There's the instrument in my lab. This is that picture from the opening. This is an example of some of the pyrites that you can find in sediments. And again, historically, we would just crush these up, dissolve them, and get one isotopic value. And instead, what we do with the SIMS is we get isotopic values for each and every one of these, and then use those distributions to tell us something about the environment. In some cases, that's not terribly useful. This is a case where the bulk value is about -44. We spent about a week to measure the SIMS value, -47. There's a really tight distribution. There's almost no difference. And so there's just a single population, a single process that's going on to create these. But in other cases, it's critically important. This is a case where the bulk value of pyrite was about +7 per mil. We analyzed all the individual pyrites, and we got an average of +7. So that's a pretty good agreement. But this is what the distribution looks like. There's a distribution of really depleted values with a little bit of a tail. This is microbial fractionation with some distillation during early pore waters. That looks almost identical to what we have in this sample. But there's a second pulse of, these are all framboids, and these are the second pulse of euhedral crystals with a value of about +40 per. These average out to +6, but there are no +6 pyrites. There's no process that gives rise to this value. This value is meaningless. It has no relevance to trying to reconstruct what happens in the environment. But what people do is they take these bulk data and they interpret them. And so this kind of approach, where you really go in and look at the small scale variability, is absolutely critical to being able to link geochemical signatures to process. And that's the kind of thing that I get excited about. And so I just want to end by acknowledging all the people that make this happen, because I spend most of my day doing emails now. And these are the kinds of people that make all of this happen. So I think with that, maybe I'll stop talking at you, and thank you for the opportunity to sort of think through how my career has progressed. Thank you.
For one of those microbial mat I guess stratigraphies, what time scale would that be on?
So the whole mat system, so one of the nice things about the Guerrero Negro system where we're working in Baja is we know that whole setting grew up over about 50 years. Because this is it's a hypersaline mat, which is the whole reason these mats exist. It's sort of an area that's being used for commercial salt collection, harvesting. And so that mat is about this thick and it grew up over 50 years. We think that the organics that go into that undergo different amounts of decay as you march up from the modern down through that. And then the sulfide that is formed is sort of diffusing throughout that. So you've got the sort of modern sulfide creation diffusing over this zone of about 50 years of microbial growth.
Thank you.
Right.
Given that, your career has had all of these serendipitous occurrences and so on, and I've had very similar experiences where fate seems to take you where you want to go, where you need to go. What's your advice to these students about, approaching a career in academia if that's what they want, or any career for that matter, on, how to be as successful as you have been?
I guess, for me, I think a very important component is maybe knowing that, I think you and I maybe both had ideas of where we would head and then where we actually headed. And so I think being open to opportunities that arise that maybe aren't part of your grand plan, but catch your attention, and then being flexible to explore those areas. And then I think for me, one of the things that has worked out really well is going from working on rocks in deep time to modern microbes. And that was a very hard transition to figure out spatial scale, temporal scale, techniques. But then you get a different sense of perspective. And then I think having that different perspective is very valuable. I think, so I guess I would say not being afraid to try things that are very different, where you maybe don't feel very qualified, has worked out the best for me. And I still don't feel very qualified on lots of these things.
We have a lot in common.
Yes.
How do you feel that you've grown as a mentor over the course of your career? And do you have any advice for graduate students that are going into academia, that are going to go on to these mentorship roles, that you could provide?
I think having a sense of perspective is very helpful. And I think realizing for me that student needs and skills and desires are different. So when I started out, my advisor was incredibly hands-off. I saw him maybe once every 6 months, and he would say, "How are things doing? Okay, good. Keep it up." And so I thought that's how you would advise students. And so I started out, and I said, "Student, go away. Come back in 6 months. Let me know what you're doing." And that works for some students, but it doesn't work for other students. And so coming up with a really flexible approach where now, like when I have students, when I talk with students about coming to the lab, you talk to me about how you would what you're looking for in an advisor. What kind of regularity? Do you want micromanaging? Do you want no managing? Where are we? And then we work together on sort of an approach, because if you take the wrong mentoring approach, it can be pretty disastrous. And so I think just for me, understanding that, we need a different approach for different kinds of students was really important. And I think the advice that I would then get is to make sure that you have these conversations with your advisors or your future employees about like, how would we interact? And these are the things that I need. What do you provide? And I think having that conversation up front can help a whole lot.
Yeah, great, thanks. Okay, well, if there are no more questions, we have a lunch. Yeah, following the seminar in Challenger in the Swanson room, we have paninis from El Sol, Mio, and baked ziti and that sort of stuff. And it's another opportunity to talk to Dr. Fike more about his research and career journey over there. Yeah, so let's give one more round of applause.